Warm greetings and a very good evening to one and all. I cordially welcome you all to the second day of the Faculty Development Program on Frontiers in Applied Biosciences and Research Innovation. A special welcome to Dr. R. Shobana Manoharan, Assistant Professor, Department of Biotechnology, Tyagarajar College, Madurai, who is the resource person for today. I now invite Dr. Janet, Associate Professor of Zoology, to deliver the welcome address.
Good evening to everyone connected here. It's my great pleasure to extend a warm and cordial welcome to our distinguished guest speaker and all the participants who have joined us today for this faculty development program through the online platform. We are delighted to have you all here for this academic session. Today's session is particularly special as we have with us an accomplished academician and researcher, Dr. R. Shobana Manoharan, as the resource person for this program. Dr. Shobana Manoharan is currently serving as an assistant professor in Department of Biotechnology at Tyagaraja College, Madurai. She has also worked as a research associate in Department of Immunology at the School of Biological Sciences, Madurai Kamaraj University, and earlier as an assistant professor at Lady Doe College, Madurai. With several years and research, significant contributions to the field of biotechnology. She obtained her doctorate in biotechnology from Madurai Kamaraj University, where her research focused on the genetic diversity and biochemical characterization of the freshwater fish Lepidocephalus thermalis. Her academic journey has been marked by excellence. She secured gold medal in both her BSc and MSc biotechnology programs from Ayanada Janagi Ammal College and PSG College of Arts and Science, respectively. Dr. Shobana has successfully cleared GATE and Transit in life sciences, demonstrating her strong academic foundations. She has published 8 research papers in reputed national and international journals, contributing to areas such as molecular biology, microbial genomics, fish genetics, immunology, and antimicrobial research. Her technical expertise span a wide range of advanced techniques, including PCR, gene cloning, sequencing, ELISA, flow cytometry, protein analysis, and next generation sequencing data analysis, along with bioinformatics tools for molecular docking and drug design. Adding to her achievements, Dr. Shobana is a recipient of the Young Scientist Award from the Indian Science Congress Association, which recognizes her significant contributions to animal, veterinary, and fishery sciences. She has also served as a technical resource person in several workshops and training programs on molecular biology techniques. With her rich academic background, research experience, and passion for knowledge sharing, we are confident that today's session will be highly informative and beneficial to all participants. On behalf of the organizing committee, I extend a hearty welcome to Dr. R. Shobana Manoharan and thank her for accepting our invitation to share her expertise with us. I also warmly welcome all the participants who have joined this FDP and wish everyone a productive and enriching session. Thank you.
Thank you, dear ma'am, for your warm words of welcome. Dear participants, we kindly request you to turn on your camera so that we can take a photograph of the second day of the FDP. Thank you. I now cordially invite Dr. P. Shobana Manoharan on the topic From Morphology to Molecules, DNA Bio to you, ma'am.
Thank you, ma'am. So thank you for providing me this opportunity and thank you for the good introduction, ma'am. So now I hope the session will give us a good insight on DNA barcoding in specific with the fish biology. So while we begin with this, before we go into the DNA barcoding, let's first start with the basics and meristic characteristics, that is the countable ones. So how many numbers are available? So based on that, we had so many classifications. So upon that, after while we were going beyond, so while you have some cryptic species to be understood, so in those cases, we wish that so some of the markers could help us. So in that case only there was involvement of DNA based markers. So directly before we go into a barcode, so first let's see what are all those kinds of markers. So this challenges made us the requirement. Otherwise, so the morphometrics was very convenient for us. But the thing is, we need expertise in it. So if it is going to be in its juvenile stage, or it is in a fry or a fingerling stage, in case of a fish, so how you classify? There were questions. So we had so many problems aroused that can be difficult for us to identify what kind of fish it could be. So this case was observed in case of ornamental fish culture, as well as while we started exporting the fish. So we have so many situations where we sought a DNA based marker. So the traditional approach, it involved the external features. So in some cases, you can see based on the fish bones, people identify what classification it could be. So here, the strength of the particular morphometric technique is it is going to be a direct observation. So we need not require any specific lab equipment like hand microscope, maybe that tool to identify very fine rays. Otherwise, we will be able to identify all those. But while it comes to the other classes, like when there is going to be environment influence over their phenotypic traits, or it is going to be between two closely related species. For example, in case of a freshwater fish, Chenna species. So you have Chenna striata, Chenna punctata. So in Tamil, we tell it as Viral and Kuravemi. So you have both species from the same genus, but while you appear with their spots, so these spots, while they become a grown adult, you'll be able to find it out. But in their early stages, either it can be through the egg or larvae. So these are very important while you go with the aquaculture. And in some cases, so now recent days we have the processed foods. Yes, so we claim that it is from a particular fish. So we do not have their entire morphological characteristics, but still we are in a position to justify whether it is going to be the particular fish. So in all such cases, we require a molecular taxon based approach. So that is why we have transferred slightly from the morphology. So it is not that morphology is not at all being done. We definitely do the morphometrics, meristics, but in some cases where we do not have such options, we serve as an essential tool. So in case of ichthyology, so basically, so how we classify the fish. So either based on their jaws, so their bone, the cartilaginous thing, and you also classify it based on the mouth position. So how its mouth position is being there, and based on that, how it is going to be a feeder, whether it is going to be a top feeder, column feeder, or a bottom feeder. So we have several classifications based on their anatomy and physiology. So while we have all these, or while we go to the field of study, so we just collect the fish sample, then we make for all these entries, then with the help of a expert. So for example, in case of a botany, we have a herbarium based preservation. So like that, here we start preserving the fish specimens along with the formalin solution. So once we examine all their morphometrics and meristics, we save them and we just give their name. This was done initially. But now, if I have some authentication, so what can I do? It is not necessary that I require the entire fish, or it is not necessary that I require their blood sample. It can be even their caudal fin tissue, a tail fin tissue that can be chopped into, or their scales. So even their scales can be utilized for extraction of DNA, and they can be utilized for their barcode study. So once we extract the DNA, then we'll check into how we study for their cytochrome oxidase 1 based gene. So before I go with the other part, let me just give you some basic measurements, whatever we do with the morphology. So whenever we get a particular fish for its morphological study, so we measure its total length, its standard length, so head length. So we have each and every measurements that is being done with a scale, classical scale. So you measure them, note them all. This is all going to come in a morphometric based study. So when it is going to be the counts, so here if I find 7 rays, 8 rays, so that is going to be a meristic based approach. So apart from this, the scale types people start record. And they also record for their mouth, whether it is going to be a terminal, inferior, or a superior. This determines their feeding behavior. And we can also see their caudal fin shape. So either it can be a truncated or a rounded. So these are some of the classifications. So based on which how we classify which kind of fish it would be. So for the classical ones, we do this. So while we found so many categories of fish, so in 2003, the DNA barcoding concept from Paul Hibbert was given a good, important, essential definition for DNA barcoding in fisheries. So how this could have been arisen? So while you check into the variation, so we have certain random markers. So like RAPD markers, then we also have ISSRs, simple sequence repeats. So even these markers initially between the fish that is being captured, which had the same morphometric and meristic characteristics. So then later people would like to know, so how far it would be beneficial so to identify them. So in that case, while we focused on the mitochondrial gene cytochrome oxidase 1 in fish. So here A for bacteria. So for a comparison, so if you take either their 16S or your RNA polymerase beta gene. So in case if it is going to be a plant, we take their maturase K or a rubisco L gene. In case if it is a fungi, we take its 18S or its ITS region. So like that, we have specific region. Why people focus on those regions? So they are going to be involved in the evolution-based genes that can help us to understand their phylogenetic relationships also. So the genus species classification can be clearly understood. So that is why we start going with those. So after that, there was a consortium for this particular barcode of life. So in specific for the fish barcode of life was initiated in 2005. These are like the databases. So like when we tell, if I study about a morphometric characteristics, I record. So in case of this fish, this can be the standard fin ray count. This can be this dorsal fin ray count. So like that, I give each and specific information which is being available, which we refer into, right? So when it became with the sequencing, so what happens here? I extract a genomic DNA. So that is a total DNA there along with I have their mitochondrial DNA. So now I have specific universal primers for cytochrome oxidase 1 gene. So this cytochrome oxidase 1 gene is amplified using a polymerase chain reaction technique. So then this is being sequenced. Once those sequences are available, so we start submitting them to a database. So Fish Gold was one such initiative in 2005. So at that time, we did not have much of their whole genomes available, but we had some of these gene-specific sequences. So later in 2006, there was a gold database that is being established. So this barcode of life database has sequences not only for fish, but it also integrated for various living organisms. So every time, whenever we make a sequencing study, so we submit those sequences to any one of the public primary databases. So the three primary databases include the NCBI GenPack, we have the DDBJ, and the EMBF. So we submit it to any one of the concern and they provide us the accession number which we use for our publications. And there is always a regular exchange between the data between these three databases. But here the whole database is quite special. So what is special over here is, so while we submit our barcode sequences over here, So there are specific barcode index numbers that are being provided from these databases, and they also have certain classification and tools that are being given for working out with these sequences. So that is why they formed a major repository for DNA barcode based sequences. So apart from NCBI, here they will have only DNA barcode based sequences. Bold itself will give us a accession number in case of a barcode index number, and along with that, it is in tie-up with NCBI where you get the NCBI based accession number. So both we'll be getting for our sequences. So then in 2008, so we had so many global fish barcoding projects that were available. So while it comes to riverine fishes and the native fishes for each of their nations, so they were being barcoded and they were being uploaded. And later, this was expanded with more and more sequences. And now we have complete meta barcoding for fish samples that are available from the environment. So now it is being sequencing with their whole genome being available. So now, as I said, so it is going to be a standardized method that uses a short DNA sequence. So it is only about 650 base pairs. So, and this is being used as a barcode. So as we all know, why we denote it as barcode is it is going to be unique for each of the genus which we study. So whenever we go for a supermarket, so these days we find n number of barcodes that is being provided in each of the particular product which we choose. So while we come to the billing section, it is just being scanned. We are able to identify the kind of product it is. So it gives the entire information. Even in certain cases, you can see till its data that is being fed. So like that, now we have the databases, repositories with enough sequences. So now you have enough number of technologies that have been available. Now, whenever we provide a sample and once its sequence is available, so while you match it along with the database sequence, we'll be able to identify what kind of fish, that is the genus it belongs to. So here, always the standard gene which we prefer here is the mitochondrial cytochrome C oxidase subunit 1. So this is because it evolves quickly for the species discrimination and it is slowly for a higher taxa. So, and always mitochondrial DNA, it has its high copy number, and due to its maternal inheritance and rapid mutation rate, people have preferred this. And while we start studying about this, so the primers which we use, they are all based on the universal primers. So generally, they are available. In some cases, while there are going to be between the two cryptic species. So in that case, we have separate tools that can help us to distinguish between those two species. But otherwise, we always prefer a universal primer that is being used for this DNA barcoding. So here, as I said, each and every organism. So for cytochrome oxidase 1 can be used for most of the eukaryotic organisms. So here, the 650 base pair is the target region. So it is being amplified first. So once we amplify, so for that, you need to extract the DNA. So once you extract the DNA, amplify it using a PCR. So after that, this is being either you can elute the sample or it can be PCR purified. So after that, we go for a normal automated DNA sequencing based on Sanger's dideoxy chain termination method. So there, once we get the sequence, we start comparing it. So when we start comparing it with the existing sequence, so we are able to understand what kind of genus it belongs to. So now from the basic one, so while I started visualizing, I need the entire fish. So I need to make all these entries. And in case if I'm not familiar with this if I'm using it for the first time, so I do not have enough knowledge about this. So I need some authentication. But now while I come to this approach, even a fish fillet or a scale or a piece, any tissue piece is sufficient for me to make this experiment. And this molecule is going to give me accuracy rate till 99. So it can be even till 100% match. So why this is more accurate? Because it is again based on the sequences, and I'm able to match it with the existing sequence. So in some case, if I have no existing data available, then how do I identify? So then you again have other closely related genus sequences that are being available, so which gives you around 60 to 70% match. In some cases, we record a new species. So how do we record a new species? So while you have to record a new species, so there should be at least 25 percentage of divergence. So if it is 99.8% or 98.9%, something that is very close, so it is going to be the same genus which is existing in the species. In some case, if I'm getting only 75%, you found it something different. So in that case, then it can be a record of new species in case of a molecular level. So we follow for preparing a DNA barcode is, so once we collect the sample, so either it can be a tissue or a film clip. So not necessary that you require a huge amount of tissue. At least 1 mg to 10 mg of your tissue is sufficient for making this DNA barcode. So after this, we extract the DNA. So here, normally, in case of fish-based tissue, so the proteinase K or the salt out procedure can be followed. So the proteinase K concentration alone should be maintained based on your sample. So based on that, it will be able to lyse the tissue very fast. So after that, we go for a PCR amplification using the universal primer for cytochrome oxidase 1. So once we amplify, we should be able to get a band of 650 base pairs. So then we go for a sequencing procedure. So after that, you compare it with the reference databases. So here again, we submit it to a database for getting the accession number. So what we follow while we are going to collect the samples. So in case if it is going to be a study that is going to involve many other procedures like bioaccumulation and so on, So people will not store them in formalin. So it will be progressed with all those tissues that is being collected. Otherwise, if it is going to be preserved, or in some cases, even if you would like to extract a DNA from a formalin preserved sample. So again, we have certain procedures that can be utilized for extracting DNA from those samples. So once the DNA is extracted, we start amplifying. So in the PCR, we have 3 essential steps that happens. So first step, it's going to be the denaturation, where the double strands form 2 single strands after the denaturation. Then we have annealing, the binding of the primers. Then these primers extend. So this cycle is repeated for 35 to 40 cycles. So after that, we get the product that is the amplified region of cytochrome oxidase 1. So now this particular product is alone sequenced. Here again, we go for a forward read and a reverse read separately. So while we go for the data analysis, you can be specific by what kind of database you're going to choose for. So in case of fish, the barcode of life database has more options, so which we'll be seeing in the upcoming slides, where you can use them for your research and analysis. So this particular website is from the Bold Systems. So it is going to be open access informatics platform. So it was launched by the University of Gulf in Canada. So it's between 2 people. So Paul Hibbert, who introduced the DNA barcoding concept in 2003, and Stephen Ratnasingham. So here, what was their mission is, they just wanted to record all those DNA based information there. So now you can see there are so many information that can be available with them, along with your vouchered specimens, metadata, images, and your sequences. So while we submit a sequence, like I said, it is interoperable with GenBank, and it also supports the other initiatives. While we submit a sequence to a database, normally a GenBank or DDBJ, usually we do not submit our sequencing AB1 file. So the AB1 file is the raw file which we receive after we perform a sequencing experiment. So that file is not usually being uploaded while we submit a sequence for NCBI. But while you go for a barcode of live database, so as they said, right? So they have so many information. So what are the information they collect? Let me just say. So while you're going to operate there, you need to give a small introduction about the lab, or you can get a project. So DBMKU was the project which I did. So while I get the particular project, I can give my sequences over there. So while I give the sequences, I have to give the GPS location where exactly the sampling was done. So, and then the sample images. So along with that, if I'm submitting for the bar, they are AB1 file. So only when after that, it will start analyzing. So I said now the sequence size is around 650 base pairs. In some cases, if I get around 200 base pairs only of quality read, if I submit to NCBI, it will accept it, stating that as partial, either through the 5 prime or through the 3 prime. You'll be able to submit it. But in case of bold, what will happen is it will accept it, but to understand it for a barcode index number, it should have a minimum sequence length of 500 base pair. Otherwise, it will not consider it for barcode index number. If there are queries related to the quality of the sequencing which is being made, again, it will be flagged. So that is the positive advantage of using these kinds of databases. So again, both bold and fishbowl, they are extremely dedicated only for fish-based sequences. So while you would like to recover more of information about these, so you can try these two databases for their understanding. So this is how it looks. So now we have the V4 and V5 versions. So as I said, you can see there is a workbench. So this workbench gives us unique tools. So I'll just list you out some of the tools that can be utilized. Like these days, we might have heard about phylogeography. So we tell that there is a geographical indication. And we'd like to know how the genetic divergence is being operated along with the geographic distance. So in all those cases, we'll have so many tools to understand based on these kind of goal systems. So currently, as of this month, there are around 19.8 million records. So while you submit to this database, again, when you have a question, so like I have 10 sequences done this year, it may be going on. I do not want to reveal this to the public. So again, you can keep it as a private one, stating that until my publication is being done, it can be kept over private for a limited time period. So there will be options for you to store your data. And again, there you can see, so the greater than 500 base pairs, how many are there? And this barcode index numbers is being given dedicatedly only for the sequences that are having a size greater than 500 base pairs. So now generally, while you compare the morphology along with the DNA barcoding, so this morphology is very well done while it is a fish with a considerable adult size. While it is very small or when it is very young, in case of aquaculture, again, we find it a little difficult. Maybe those who are very experienced with that, they'll be doing it very well. So, but DNA barcoding is successful in those cases again. And in case of cryptic species identification, so here you can have one example for the Coregonus and the Castomus. So there, these kinds of studies are well studied with a molecule, DNA based study. So, on time, the morphology based study is quite time intensive. And while it comes to the PCR sequencing these days, even we have certain instruments that can do your on-site based recording. So like we have mini DNA barcodes. So DNA barcode 650. Within that, we are specific for what I'm learning. So mini DNA barcodes are also on their emergence. So that makes them even more faster. So, and some of the examples, so which are being resolved, like the larval fish that is available from the Lake Huron. So there, how many can be identified based on morphology was 94%, and others, the unidentifiable ones, were best resolved through the DNA barcoding concept. And in case of their advantages, so the reproducibility and the cryptic diversity, again, you can find so many case studies. Again, we'll also see some case studies from India too. So where we start applying these. And in case of authentication, these days food security. So we all claim like certain food or being priced higher, stating that it is a salmon from particular nation. So in that case, we need the authentication. So for that, the whole fish is not available, but still you have a fillet. From that, whether we would be able to do. So for that, DNA barcoding plays a vital role. And how it complements. So integrative taxonomy. So this is the thing which I would like to stress. So when there is an option, try to go for integrative taxonomy so that it could give you both a best results based on your morphology as well as the DNA based information. So here the advantages just include accurate species identification, cryptic species identification, even degraded samples can be analyzed. All stages from the egg, you will be able to do the rapidness, and it supports the biodiversity assessment. Yes. And in case of food authentication, as I said, we claim. So in some cases, we claim that this chocolate is made from a Ghana cocoa. So how you authenticate whether it is from Ghana? So for that, we do so many studies. So that kind of authentication can be done, and there is a standardized global database that is being established, and it also helps to support the conservation biology based information. So there are also certain challenges and limitations. So what are they? So sometimes, in some cases, the primers might not contribute for you for a universal study. So that may happen very rare. And in some cases, while you compare it with your nuclear as well as the mitochondrial DNA based issues. So again, the copies and their integration may do that. And database gaps. So every time the people should come forward. So only when we go for a publication, we go for submission of their data because that it's mandate. Otherwise, if you are reluctant to submit it to a database, so or the updation of the database, that may cost up. And the cost-based thing, yes. So the initial setup, setting up a PCR, standardizing them, it takes a few studies for a limitation, and the infrastructure for a portable lab. So like doing them all to be very fast is again a question mark in some places. And again, while you come with the ethical and the practical concern, so while you have to do with the voucher specimens. So I said, if I'm collecting a sample, I go directly with a DNA based study alone, and I argue that this is going to be the case. So still we are not ready to accept it. So the integrative taxonomical approach is being supported. So for the case studies, so within, let's first go with the abroad, then we'll go with the Indian-based studies. So in case of Great Lakes and Taiwan, and again, for a North Sea, we have able to record all the species which morphology could not answer. And the undescribed lineages in Congo Basin has been done, and regional efforts for Philippines, Iran mangroves. So there, the African fisheries have recognized so many fishes based on barcoding approach. So in case of India, so fish diversity from the Bees River system for Indus, so we have able to do, and catfish, so for Clarius batrachus and Clarius garipnus, so their variations was well studied. So the DNA barcoding from Gara fish in Rajasthan for Aravalli region is being done, and ornamental fish from the Moti River in West Bengal is also being a successful approach. So these applications for authentication of geographical origin, identification of a new or endangered species, validation of a food product, so corroboration of closely related and ambiguous species in case of a ornamental fish trade. So these days we have several imported pets and other fish that are being imported or exported. So for that also again we use these barcodes. Then fish diversity in a location or species discrimination study, mislabeled food products or smoked food products again. So things which are being overpriced with your name. So for that, all classifications, we employ these. So now I would like to share a few outcomes from my research, which I had made during my PhD with these fish. So first, as I said, so my research was based on Lepidocephalus thermalis iramine. So here we have collected the fish species from different locations. So we choose around 11 locations. So from there, we took fish species. Then we started. So before we went into the 650, there is a story why we did DNA barcoding. It did not happen all of a sudden because while we learned about those fish, so we had so many questions stating like this species is so popular in Madurai. So, and it has a high socioeconomic background costing around rupees 2,500 per kg. So, when it is so costly and it's available here, does it have any uniqueness? So, what we thought is we started sampling from different regions. So, once we sampled, what we did is we extracted their genomic DNA. So, here for this fish, the caudal fin was sufficient. So if you just cut the caudal fin, the fish will not have will not lose its life. It will not have any damage. So if you take a small piece of its caudal fin tissue and then you go for a DNA barcoding, so you will get
Voice is not audible, ma'am.
Dear ma'am, please unmute your mic. Ma'am, can you hear me?
Shobana, ma'am, your mic is on mute, ma'am.
for a random marker. We found there. Yes, ma'am. Is it audible?
Yeah, I think you can go a bit far. We were
Sorry?
not able to hear you for a few seconds.
Now,
Yeah,
okay.
now it's audible.
Okay. Yeah. So what I was trying to tell is, so from 11 sites, we collected the fish samples. So then we went for extraction of DNA. So the caudal fin tissue, while you clip it from the fish, it will not lose its life. It will be able to survive. So after that, while we extracted, we just wanted to know whether does it make any difference between the 11 sites, because this fish is common. While you see the morphology, there were no much variation. So it is a very small fish. Maximum it will go for around 7 or 8 centimeters, the maximum I'm telling. So it will be very small even in its adult stage. So at that time, we thought we'll check for a RAPD based study. So while we just checked through it, we were able to find some differences with their amplification. So there were polymorphic patterns. So then we thought, why not we try with a DNA barcode? Does it show any significance? So for that only, then we went with the amplification for their cytochrome oxidase 1 gene. So here we found a 650 base pair amplicon. So as I said, this is a universal primer. It was not designed. It is a universal primer. We employed it and we amplified it. After that, we obtained its sequence. So once we had the sequence, we submitted it to the BOLD database. So as I said, it is integrated along with their GenBank. So interoperable. So this is the GenBank accession number which is available for the species. Apart from this, we obtained the barcode index numbers. So these barcode index numbers are the unique numbers that are provided based on their genetic variations in their cytochrome oxidase 1 region. So now we found that the regions from Madurai had a different number. So from Theni, there was difference. From Tirunelveli, it was different. So the other ones, so the out samples which we took from Thanjavur, that is from Kaveri, why it was not given is it is less than 500 base pairs. This is what I was mentioning. So when the sequence is less than 500 base pairs, we will not get any barcode index numbers. So once we got the barcode index number, then it helped us to understand, yes, so there was variation between their genetic appearance. So now we started understanding their accumulation curve based on their sampling efficiency. So these are all like, these are the tools available over in the BOLD workbench. So from the workbench only, we are able to get all these tools. So from that, we start working and we understand. So whether the sampling efficiency is really good. So here you can see the red one is going to be the barcode clusters. It's rising. And the species, I recorded only one species, Lepidocephalus thermalis. So that is why it is with the same one. But the clusters is for 11 count. So after, yeah. Then you also have the sequence divergence analysis. So within the 650 base per region, so how the variation is. So we also had one more study between the Chenna striata and Chenna punctata. So there you can find the regions that are very specific, that is going to be a variable region or the conserved one for these two species. So then it can help you to design primers for identifying the difference between these two very clearly. So then, how many unique barcode index numbers were recovered? So they were classified and the count numbers for this project was given. So the last one, the geographic distance and genetic divergence correlation. So this was based on mantle test. And why I'm highlighting this is the one that actually helps you to give the information for the phylogeography study. So there are so many phylogeographic studies. In case of freshwater fish species, so you can see certain Malaysian-based studies where they get from like the cheddar species from one different other lake, how they migrated to the other place. So how was the geographical migration? So many studies are being initiated. So here you can see it compares the genetic distance along with the geometric distance. So based on that, it calculates the Mantle's p-value. So here it was significant, stating that they had phylogeographic based association. So the genetics varies based on your geographical distance. So once we had the 11 particular sequences out of our study, so we made a phylogenetic tree for the same using bold alignment and the Kimura to distance parameter, where we found different clusters based on their river systems. So then we would like to compare it. So this was 11 out of our study. So this 4 was other study from a Thoothukrin based Fishery Institute. So again, they had also made this. So these two are the clusters direct from Lepidocephalus. So the other all are from belonging to the Cobitidae family. So the Cobitidae family, we recovered so many other sequences counting to around 50 to 100. So then we compared all those sequences for this phylogenetic tree association. So we were able to find 6 significant variations. So based on this, after so many studies, we were able to understand that integrating both the classical and molecular taxonomy will support us to have a more effective data. So the future prospects, as I said, many barcoding, shorter fragments are being concentrated, meta barcoding for bulk samples, and NGS-based integration, environment DNA. So all those are being done. So the global goal, there are like the fishbowl databases. It should be library for all submissions, and it has around 34,000 fish species based records. So now if we visit Barcode of Life database also, you'll be able to see so many data, even for regional and other international fish. If you're trying to search also, you'll be able to find those information, including from all their genetic and their geographical based information. So out of our study, we were able to papers. So the first one was with Indian Journal of Experimental Biology. So where we had the morphological and molecular characteristics along with their DNA for both. So that is why this technique proves to be a more supportive one. And we also had one other publication based on phylogeography for Chenna striata and Chenna punctata. So again, this was based on 3 different river systems. So from Kaveri, Vaigai, and Tamarabarani. So the samples were from different resources. So from which we try to compare their barcode regions. So I'd like to give a summary stating that so the DNA barcode is going to be a shift from the morphology to the molecules. It is not replacing, but it is actually enhancing the traditional ichthyology. And it enables accurate as well as scalable identification. So these days you might be like if you tell something new, so most of the time, if the person is not in the field or if they have not handled a fish earlier, so they'll have count like how to count, from where to start. We have so many questions. But while it comes to a molecular taxon, so we have certain kits that are being framed these days that help us. Again, for morpho also, we can try to make certain databases, or once you give a image, how to make it understood. So how AI can support us, that could also be a beneficial one. And it is critical for biodiversity discovery, conservation, and for sustainable fisheries. So the best practice which we would like to follow throughout us, the integrative approach where we go along with the morphology and the molecules. But we have now advanced in molecules with more of technology. So this technology can also be tried to improve along with the morphometric tools. So the take-home messages like integrate classical morphology with the modern molecular tools. So with DNA barcoding, so you can understand the complete one. So this is just like while we have classes. So if you would like to make it interesting, so for students, we right. So here it can be in oceans deep. So where fish do roam, DNA barcodes bring them home, and it's a snippet small, a genetic key that unlocks the fish identity. So no fins confused, no scales amiss, each fish code we can't dismiss. So next time when you see a fishy face, remember you can solve the case using our DNA. So DNA can also support. So to acknowledge, I thank the college management for giving me this valuable opportunity to present this lecture. I appreciate the organizing committee for their coordination and efforts in arranging this session. Thank you all participants for your enthusiastic participation and active involvement. So thank you. Any questions? I would like to take the questions. And so this is like just for a quiz. So if you would like to play something interesting, you can just go for a play. If you just scan this QR code, you'll get a quiz which you would like to play. This is just like to recall what we learned. Yes, ma'am. I had someone raised a hand. Any
Ah,
questions?
Good evening, ma'am.
Yes, ma'am.
This is Luth Mary from Lady Do College.
Yes, ma'am.
Thank you for a very insightful explanations that you have given.
Thank
I
you,
have
ma'am.
one clarification.
Yes, ma'am. Yes, ma'am.
The primer that you have used is a universal primer.
Yes, ma'am.
Okay. Another thing is you have morphological for the fish,
Mm.
and then you characterized the molecular.
Yes, ma'am. Yes.
Okay. So does it really complement each other or any discrepancies that you encountered in your study?
It complemented each other. The thing was,
Okay.
what I'm telling is, like, the complementation was, it was Lepidocephalus thermalis from all the 11 sites. There were no specific variations for this. But while you go with the molecular, their variations was at least 2 to 3% variations was there.
Okay. Do you think that in your study were you able to encounter any cryptic species within that species that you have been using?
In my study, no, actually at that time. Maybe if we go for even more sampling,
No.
maybe we could have got.
Mm, okay. One last
Yes,
question, ma'am.
yes, ma'am. Yes, ma'am.
How many sequences you would have done to characterize? Just 11, or you would have taken more than 11 samples?
No, no. At that time, we did only 11 only, ma'am, because whatever we found it successful through a random marker, then we shifted it to this. So
Oh,
it was totally only 11 only we did.
okay. So we need to make a note of it that
Increase
our
the sample
amplicon
size.
should be more than 500, right?
Yes, yes, ma'am. If it
Okay.
is less than 500, GenBank's accession number, we'll get it,
We can get.
definitely. But
But
R2 index number, you will not get them.
okay.
And the AB1 file, you should receive it, ma'am, every time. In
Okay.
some cases, because these days, some people send the sequence only through a PDF file, they don't send the source file. But here, definitely, you have to upload the source file.
Course file. Okay.
Yes,
One more curious question. You used
yes,
because scales and small tissue from the
yes,
fish
yes.
without sacrificing it. You have used a kit method or a conventional method of DNA isolation?
Conventional method. Conventional method we used, ma'am.
It's really nice to see your gel photos, all bright, showing the
Yes, ma'am.
same. Thank you, ma'am.
Yeah. It
So,
was conventional method only. In conventional method, that is what the proteinase key concentration alone, based on your sample, it will vary, ma'am. So for
okay.
caudal fin tissue, the proteinase concentration which we used was standardized in our lab. It was nominal, but while you use a muscle tissue, it will vary again. So based on
Okay.
that, once you standardize, you will get a very good result, ma'am.
Did you use RNA also, ma'am? RNAs?
No, like RNAs for removing the RNA contamination. Yes.
Yeah.
I thought you are telling about a RNA study because I did not do that.
Okay. So you haven't encountered any condition. Just
No,
your
ma'am.
protocol
No.
is working with the DNA very good, I think.
Yes.
Thank you so much, ma'am. Thank you.
Thank you, ma'am. Thank you.
Good evening, Shobana ma'am. It was really
Yes,
very
ma'am.
wonderful, ma'am.
Thank you.
The session was very informative, and I have very basic question, ma'am.
Yes, ma'am.
Might be very
Please.
silly also.
Any
Actually,
no
you
issues.
said that
Please, ma'am.
this
Mm.
species is very costly, and because
Yeah.
of that, you have tested in different areas like Madurai, Theni, some
Yes. Yes.
places, right?
Yes. Yes. Yes.
So why you had that difference, ma'am? When you identified, why you had that
Ah.
difference in the sequencing, ma'am? Can you say any species difference, or what is the reason for that difference?
Okay, the reason, like what we are trying to, what we thought it could be is because we tell that it is authentically very tastier in this region. So it is, while you search for its geography, while you go towards the north, you'll not be able to find these species. So in Sri Lanka, you can find these species. So while it is found in different regions, so whether like we have the same cocoa from most of the places. Even Kerala has cocoa here. But while we have Ghana, we tell that the taste is quite different. So it is costlier. It is making the uniqueness. So in that aspect only we thought, because the genetics, maybe the environment, the geography supports it. That is what was our question, ma'am. Maybe due
But
to
there is
the
no species difference, right, ma'am?
no,
The
no
species
species
is not
different.
in any way, it is not different.
Not different. It's not different, ma'am.
In
Because
Tamil, it
the
is called as
record is
Airai, right, ma'am? If
yes,
I'm
ma'am. It
not
is called
wrong,
as
in
iron.
Tamil, it is called as Airai,
Iron.
mean.
Iron,
I
ma'am.
okay,
Iron.
okay. But now it's endangered, no, ma'am, endangered, and it is in a threatened status only.
No, it is least concern only. LC, least
Okay,
concern.
okay, ma'am. Fine, ma'am. Thank you
But
so
it
much, ma'am.
is
It was really
welcome,
very
ma'am.
wonderful session. Thank you.
Thank you, ma'am. But it is found in specific regions only. It's not in
Yes, very
endangered.
specific
It is
region
found.
and very limited supply also. It could not be cultivated. There is very more difficulties in cultivation too.
People are cultivating in tangible beds. Again, there are certain pond-based cultivation systems going on. Some
Okay.
people do. Some people are still doing on it, working on it now. Even the fisheries
Not
colleges.
frequently, right? Like not other
Yeah.
aquaculture processes. This is not in a very um, possible
Yes,
way.
yes, ma'am. Because of the feeding habit of the fish and its survival again. So it is very good in surviving in a swampy mud region based things. So it likes the river flow to be on. So it has so many specifications for its survival. So that makes it
Yeah.
different. You cannot
Yes,
cultivate it
ma'am.
in a tank very easily.
Yeah, understood. Thank you,
Yes.
ma'am. Thank you so much.
Thank you, ma'am.
Evening, ma'am.
Yes, ma'am.
This is Dr. Nalini
Yes, ma'am.
from Pumhar College near Mailadurai.
Yes,
Uh,
ma'am.
I have a basic question, but I think this is where you can upload your sequences to NCV as well as bold, right? That is only
Yes,
because
ma'am.
of the
Mm.
size of the base pairs, right?
Mm.
650, you said it should be more than 500.
500. Yes, ma'am.
And you told that if it is going to be more than 500, if I have
Mm.
two sequences,
Mm.
the same species,
Mm.
is it that I can load to bold or I can submit in NCBA?
Ma'am, actually, it is like while you go to the bold itself,
Mm.
two are from different leprosaurus. See, like if you see here, okay, so here all these 11 are from Lepidocephalus only, but different sites they were connected. Okay, ma'am. So
Mm.
now I got this,
Hmm.
I submitted to bold only. So while you submit it to bold itself, it will link for a GenBank accession number for the entire thing. But for this alone, it will consider only greater than 500.
No, I'm not asking that actually.
Okay.
Actually, you said you have collected one Delhi and Teni.
Yes.
I can keep it like
Yes.
that, like more than
Okay.
500.
Okay.
So if
Okay.
you submit both to bold, you get
Yes.
accession numbers.
Yes.
It will be two different accession numbers, right?
Yes. Yes.
But NCBA, can you submit the same?
No, this is
Instead of
the
bold,
ma'am. Okay. Okay.
instead of bold, can I submit it to NCBA?
Okay. If you give bold alone, you can go and submit it to NCBA with two different ones.
Okay,
If
so
you
what?
submit directly to bold alone, you will get only this index number, ma'am.
Okay.
This index number can be given for some other person also. For example, if you are working with the other species in Teni, the same iramine, while you are getting the different accession number, this conserved region, if it is going to be the same, again I'll get the same. So for example, here you can see for all these three, I got only the same barcode index number. That means the
See,
particular region which they chose was the same.
okay. Okay.
Yes.
But this gene accession number is from NCBA, you mean, or from board? Gene
This
accession.
accession, this is from Bold. Sorry, this is from NCBI. The last one is from NCBI.
Okay. That's what I had it out. Because we submitted to using CY gene, we partial sequence only we submit.
Yes.
But since you said that there is a barcode index number, so I just want to know about that. I have some more clarification on that. Can I have your mail ID for that?
Yes, ma'am. I'll just give it in chat window.
Okay, ma'am. Thank you very much. It was a very nice presentation.
Thank you,
Thank
ma'am.
you so much.
Thank you, ma'am. Did I miss any question? If there are any questions, please do let me know.
Thank you, Dr. Shobna. That was a lucid presentation. Very beautiful.
Thank you, ma'am. Thank you, ma'am.
Thank you so much, ma'am, for sharing your knowledge and expertise
Yeah.
and for spending your valuable time with us.
Thank you, ma'am.
Being grateful is the fairest of all virtues. I invite Dr. Selvi, Assistant Professor of Zoology, to propose the vote of thanks.
Gratitude opens the door to the power, the wisdom, the creativity of the universe by Deepak Chopra. A very good evening to one and all connected here. I deem it a great honor and privilege to propose the vote of thanks. Let me first of all start by giving glory to the Almighty God for making today's program a resounding success. First and foremost, I thank our resource person, Dr. Shobana Manogaran, Assistant Professor, Department of Biotechnology, Tyagarajar College, Madurai. Thank you, ma'am, for your excellent presentation on From Morphology to Molecules, DNA Barcoding in Etiology. It's a very useful and thought-provoking session. Thank you, ma'am. I would
Thank
like
you.
to thank our secretary, Reverend Sister Dr. Pulande Teres, Principal, Reverend Sister Dr. Jessie Fernando, Deputy Principal, Reverend Dr. Sister Elil Arasi, SSC Director, Reverend Dr. Sister R.O.K. Genesius Alphonse, and Controller of Examinations, Reverend Sister Rita, for your continuous encouragement. My heartfelt thanks to our head of the department and the faculty members. My special thanks to all the participants from various institutions. Thank you, one and all.
Thank you.
Thank you, ma'am, for your words of gratitude. Before we wind up the session, I request all the faculty members and the participants to kindly fill the feedback link. The form has been put up in the chat box. I kindly request all the faculty members and the participants to fill the feedback form. The link is being put up in the chat box. Thank you, one and all. Let us virtually meet again tomorrow to enhance our knowledge. Once again, thank you.
Thank you, ma'am.
Ma'am, please share it in the group, ma'am.
Sure, ma'am, the link will be shared.
Thank you.
you.